Now, welcome to Lipid Chemistry Part 2nd, where we will discuss the classification of lipids, into simple lipids, compound lipids, derived lipids, and precursors. This group is a miscellaneous group of lipids. So first of all, simple lipids, which are classified to oils and fats, and the second group is waxes. The first group, oils and fats, and first of all, we can go through it. And fats and oils are they are structurally triglycerides, and they are formed of glycerol and fatty acids or glycerol esters of fatty acids. And triglycerides are the best storage form of energy, like glycogen. But on comparison, it's always the triglyceride is the best form of stored energy, because glycogen is having lots of hydroxyl groups in them because it is formed from glucose. It's highly polar and attract lots of water molecules, unlike triglycerides, which is non-polar, and anhydrous do not attract water molecules. So glycogen occupy lots of spaces, unlike TG, and so glycogen is it's not a very concentrated form of stored energy, and triglyceride is the best storage form of energy. Now, this is the basic design of synthesis of triglyceride as well as hydrolysis of triglyceride. I can see here glycerol takes up one molecule of fatty acid to become monoacyl glycerol. So whenever we mention monoacyl glycerol, we have to remember that it's the second carbon of glycerol that is occupied with the fatty acid. So second, it is actually 2 monoacyl glycerol. As well as here also, the last when triglyceride is broken down, the last fatty acid to leave the original triglyceride is the fatty acid that is attached to the second carbon. So here it is first of all monoacyl glycerol plus 1 fatty acid, 2,3-diacyl glycerol, and one more fatty acid becomes 1,2,3-triacyl glycerol, and breakdown is just the reverse of this process. And one more thing we have to remember or have to note here is that triglyceride here. So this is actually the D isomer of triglyceride, because the second fatty acid is attached to the second carbon is on the right side. So this is a D isomer of triglyceride, whereas the L isomer will have this second fatty acid attached to the left hand side of this carbon atom. So there is an isomerism, L and D isomerism in triglyceride, and what is shown here is the D isomer of triglyceride. Now, triglycerides are neutral fats because they are not charged, and they can be simple homoglycerides or mixed heteroglycerides. Homoglycerides, when the 3 fatty acids attached to a glycerol are of the same type, like in the case of tripalmitin when 3 palmitic acid, or triolene when 3 oleic acid are attached to a glycerol molecule, glycerol molecule. Whereas here 1,3-dipalmitoyl 2-oleine means the second carbon will have an oleic acid, and first and third carbons will have palmitic acid attached to them. Now, functions, we have discussed some of these functions, like it acts as a reserve food material, insulation, padding material, then food additive. All these we know, or we have discussed them. One of them is insulation. One of the points that we have to mention here is that in animals which live at cold climates or cold waters, they will have a subcutaneous layer or pad of fat named blubber, which help them, which protect them from low temperatures. So that is one of the functions of triglycerides or fats, in addition to the functions we already discussed. Now, this is physical properties of oils and fats are compared. Oils which have actually they are of plant origin, they tend to be liquid at room temperature, and this is because they are having more number of unsaturated fatty acids in them, and unsaturated fatty acids have low melting point. So that's why oils tend to be liquid at room temperature, whereas fats, which are originally they are of animal origin, fats, they tend to be solid at room temperature because they dominate in the presence of unsaturated fatty acids, and unsaturated fatty acids have high melting point. And there are soft fat and hard fat. Soft fat means they have, in comparison to hard fats, they will have soft fats will have more number of unsaturated fatty acids, whereas hard fats will have lesser number of unsaturated fatty acids. And fat constants, these are some values, some calculated values of fats with which we can compare different types of fats of different origins. And it can be used for identification of fats, detection of adulteration in stored fat, and determination of the biological values of given fats. So they are the fat constants here mentioned are saponification number, this one, yes, acid value, iodine number. We can examine one by one. So saponification and saponification number at first. Saponification is the process of boiling fat with the alcoholic solution of strong alkali like NaOH or KOH when the fat or triglyceride give glycerol and soap. Obviously, fatty acid along with the alkali like KOH or NaOH gives soap. So fats, phospholipids, glycolipids and waxes, they are saponifiable because they contain fatty acids in their structure, whereas steroids are not saponifiable because it is not having fatty acid in their structure. Now, saponification number is the number of milligrams of KOH. It is milligrams of KOH which is required to saponify 1 gram of fat. It is 1 gram of fat. Now, saponification number in human fats, it is 194 to 198, whereas in coconut oil, it is 253 to 262. And obviously, fat is having a lesser number of saponification saponification value. And the saponification number is actually inversely proportional to the molecular weight of lipid. And fats, the fatty acid present in fats will have a greater molecular weight because they are long chain fatty acids. And the oils will have a lower molecular weight for the fatty acid because they are having a short chain fatty acids. So this is the reason why saponification number is inversely proportional to molecular weight of lipids, and human fat is having a lesser saponification, while coconut oil or oils generally have a higher saponification number. Now we move to acid number. The definition goes like this: the number of milligrams of KOH required to neutralize. It is neutralize, not to saponify. This is to neutralize the free fatty acids. Again, this is free fatty acids of 1 gram of fat. So the definition is much similar. This also milligram of KOH to 1 gram of fat. Here also milligrams of KOH for 1 gram of fat. But this is not to saponify, but to neutralize the free fatty acids. It's the free fatty acids already present. So we are not actually breaking down the fats or breaking down the triglycerides. It's the free fatty acids which are assessed. And this actually shows the deterioration of the stored fat, stored food. And the number indicates the degree of rancidity of the given fat. Rancidity means the deterioration. We will discuss the rancidity in the next slides. So this is acid number. It shows the deterioration of the stored fat. Now we move on to iodine number. The definition goes like here. This here, the number of grams of iodine taken up by 100 gram of fat. So that is grams and grams. Grams of iodine, 100 gram of fat. And this actually indicates the degree of unsaturation, which means that if there is greater number of unsaturation, the greater is the iodine taken up by double bonds or double bonded carbons. So greater unsaturation mean for butter, the iodine number is less. Butter, obviously, it is of animal origin and it's a fat, whereas the oil, sunflower oil, is having a higher number of iodine number because it is having higher unsaturation. So obviously, iodine number indicates the unsaturation of the given fat. Now we move over to rancidity of fat. Rancidity is basically, as I said earlier, it indicates the deterioration of the stored food. It actually imparts unpleasant smell and taste to the stored food, stored fat. It can be two types. It can be of two types. One is oxidative rancidity, the other is hydrolytic rancidity. Oxidative rancidity, when oxygen is present, it is a sort of auto-oxidation induced to the stored fat. Whereas hydrolytic rancidity, because actually there is no air or oxygen involved, hydrolytic rancidity is because of moisture and because of some enzymes also. Enzyme means it may be due to microorganism, contamination by microorganism, or it may be due to poor storage techniques. So anyhow, this can indicate the poor quality of or poor taste and smell of the stored fat. In fact, there is a process involved in rancidity, the mechanism of rancidity. The first step or the initiation step is whereby molecular oxygen combines with unsaturated fatty acids, producing hydroperoxides and peroxyl free radicals, both of which are highly reactive and unstable. And it moves on to the second stage, propagation. And this is when these unstable byproducts mentioned earlier, it reacts with other lipids also. So that is a further propagation or of deterioration. And final stage is termination, marked by production of certain unreactive compounds like amides, alcohol, aldehydes, hydrocarbons, ketones, all of which will deteriorate the quality of the stored fat. So this is what happens during rancidity of fat. Now we move on to waxes, which come under as a second category of simple lipids after oils and fats. Now, waxes are esters of higher fatty acids with non-glycerol, high molecular weight aliphatic alcohols. One example for this alcohol, for such type of alcohol, is myricil alcohol. Generally, alcohols having a carbon number of C14 to C36 are chosen. And fatty acids also, very long chain fatty acids like a carbon number chain length C14 to C36 are chosen. So waxes are generally hydrophobic, resistant to oxidation, and there are examples like beeswax given here. Beeswax, we know it's chemically or structurally they are a mixture of myricil palmitate, that means myricil alcohol plus palmitic acid, then free fatty acids, alcohols, all these form beeswaxes. And carno wax is that is actually leaf cuticle. So that is forming a covering over a number of plant leaves, or it is generally called plant wax also. Now, sperm whale oil, it is present in the head of sperm whale. Almost 75 to 90% of the head of a sperm whale is formed of wax esters. And these wax esters, that means wax plus fatty acids. So these wax will solidify under lower temperatures, making it easier for the organism to dive deep into water in search of food. So that is advantage for a sperm whale because this wax solidifies and add to the buoyancy of the organism. But unfortunately, the organism, this whale, was hunted in olden days for the plentiful oil that was present in it. Now, there is another example, lanolin. It is wool fat. It's a protective wax coat over wool fiber. Now, this lanolin takes up water without dissolving. So that is the specialty, and this character is being used in pharmaceutical industry because lanolin is used as a base in ointments. Now we move on to compound lipids, the second category of lipids. Now, under which phospholipid, glycolipid, sulfolipid, and lipoprotein are included. Now, out of which lipoprotein will be dealt in a separate session. First of all, we can go to see the phospholipids. And before that, this is what we have completed here: simple lipids, and we have started complex lipids. And this is phospholipids, the glycerophospholipids and sphingomyelin. Now, phospholipids are derivatives of phosphatidic acid. We can see what phosphatidic acid is. And they are amphipathic molecules also, which means that they got polar and non-polar ends in it. And basically, phospholipids form the integral part of a plasma membrane. As we can see here, the double lipid membrane, the double membrane here, this is phospholipid, along with proteins and cholesterol and monoacylglycerols. and this is lipid bilayer, and this is a figure that represents a phospholipid with a hydrophobic head and a hydrophilic tail. Now, this is the structure. Now, phospholipids are divided into glycerophospholipids and sphingomyelin. Now, first of all, glycerophospholipids. The basic plan of glycerophospholipids is phosphatidic acid, which means it will have a glycerol, the alcohol part. Two of the carbons will be attached to two fatty acids through dehydration synthesis, that is loss of water molecules. And the third carbon is attached to a phosphoric acid molecule, phosphate molecule. Now, in glycerophospholipids, this phosphate will have an attached alcohol. And depending on the type of alcohol present in this part, it can be named, it will be classified, and it will be having different functions also. So like choline, ethanolamine, serine, or inositol, all these alcohols can get attached to this position. So that is the basis of classification of glycerophospholipids, and based on that, we can have phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, phosphatidylnositol, and so on. So these are the most commonest of the glycerophospholipids. Now, first of all, phosphatidylcholine, which is also called lecithin, it is better known as lecithin. This is the most abundant phospholipid of the cell membrane, and on hydrolysis, it can yield the subunits of it, namely glycerol, fatty acids, phosphoric acid, and choline. And this can form citrion having acidic phosphoric acid on one end and a basic choline at the other end. So these both of them are present in the same molecule, so it can actually form citrions also. And these are the functions. Yeah, this is again. Yes, these are the functions shown here. It's highly polar, so can orient along plasma membrane. Obviously, all the phospholipids are there in the plasma membrane. Significant role in structural organization of protoplasm, as well as the membrane formation of cellular organelles also, cell membrane. Then represent, this is a unique function for lecithin. It represents the major storage molecule of choline. And choline is it is important in nervous transmission as acetylcholine. So acetylcholine, the precursor of acetylcholine is choline. So this is a major storage form of choline also in our body. So the structure, regarding structure, so this is the choline part. Choline is actually like this. This OH group with the loss of a water molecule, this forms phosphatidylcholine or lecithin. And one of the curious points is that the snake venom contains a particular protein, an enzyme, lecithinase, which converts or breaks down lecithin to lysolecithin. And lysolecithin is responsible for hemolysis or breakage of RBC in snake victims. So that is one point. Now, another glycerophospholipid is phosphatidylethanolamine. The structure is quite similar to that of cephalin, and this is called that of lecithin. So this is called cephalin. This is cephalin. Cephalin structure is like this. This choline is replaced by ethanolamine. Ethanolamine looks like this. This is the structure of ethanolamine and will be replaced here with again loss of a water molecule. And what are the features? It is structurally similar to lecithin. Cephalin is structurally similar to lecithin, as we saw. Choline replaced by ethanolamine. It can also form citrions. We can inspect what a citrion is here. Like here, phosphate group is having a negative charge, and the choline is having a positive charge. So that makes it a citrion, just like in the case of phosphatidylethanolamine or cephalin also. Here negative charge, and this is the positive charge. So that's obvious there. And what are the functions? It's quite similar. It forms the integral part of all the membranes, like plasma membrane, cellular membranes, the membrane of cell organelles, protoplasm. But this is especially abundant in brain. So that is one unique feature of cephalins. So, and another one is phosphatidylinocytol. Phosphatidylinositol will be like this. So this is an isomer of inositol that gets attached to the phosphate group here. It is called myoinositol, and you can see minor rearrangements being the isomer of inositol. There are minor changes at the third and fourth positions. So it is actually myoinositol which gets attached to the phosphatidic acid group. And what are the features? The precursor. Oh yeah, this is the most important point here to be remembered. This acts as a precursor of second messengers because phosphatidylinositol, it can be cleaved to diacylglycerol and inositol triphosphate. Both of them act as second messengers of hormones, and we know what second messengers are. We will deal with second messengers in another session. Now, phosphatidylinositol is an important constituent of cell membrane, as all phospholipids are. And here serine gets attached here, another alcohol. This is actually OH group containing amino acid. We know serine, what serine is. So that also does the same thing like here. It can also act as a zwitterion. And there are other phospholipids also, which, for example, cardiolipin is one of them. Phosphatidylglycerol, that's the major lipid of inner mitochondrial membrane. That is important. And the structure is here. So this is the structure. And another one is plasmalogins. Plasmalogen structure is here. So this is a curious structure. You can see that already there is an ethylamine. So it's a basic structure or basic plan of cephalin. The second one we studied among glycerophospholipids, cephalin. But in addition, there is this portion also. It's an ether linkage, whereas this one is an ester linkage, that is between acid and alcohol. This comes as an ether linkage. Now, ether linkage is an oxygen connected to two alkyl groups. So that's what makes it different. Plasmalogens make it. The structure of plasmalogen is unique because of this ether linkage. It is similar to glycerophospholipid in structure, but possesses ether linkage on the first carbon. So these compounds constitute as much as 10% of the phospholipids of brain and muscles. So that is important point to be remembered. And we move on to sphingomyelins or sphingomyelins, which comes under phospholipids. The first one was glycerophospholipids among phospholipids. Then this is sphingomyelids, which is also called sphingomyelins. And as you should remember, the glycerol part is replaced by sphingosine or sphinginin, as you can see here. So sphinginin is having two sides or two spots, one of which will be occupied by fatty acid, the other by like phosphoric acid, with the attached choline also. So this is the structure here. Yeah, this is the structure. Sphingosine, fatty acid can see here, and the phosphate group through which a choline is attached. But basically, this is having the alcohol sphinginin or sphingosine. Now, sphingolipids, derivatives of sphinginin. Sphinginin is long chain unsaturated nitrogen containing amino alcohol. That also you can see here. Yeah, nitrogen containing alcohol here. And sphingolipids contain sphinginin, phosphoric acid, fatty acids, and nitrogenous base. And the base commonly seen is alcohol, and mostly it is choline. And what is the example? Ceramide. Ceramide is the best example. Ceramide form essential constituent of myelin sheath. So it's also called sphingomyelins. Sphingolipids are abundant in blood plasma, RBC membrane, brain and nerve tissue. It forms the structure in myelin sheath also. It has a role in the plasma membrane. So just like other phospholipids, it has got varied roles, as we can see. And then goes to glycolipids. So glycolipids, as we can see, the simple lipids are over, phospholipids are over, and we move on to the complex lipid, the glycolipids, which come under under the complex lipids. Okay. Now, Glycolipids, obviously, they are similar to sphingospholipids in that they have the sphinginin or sphingosin as the basic alcohol in it. But attached to it, apart from a fatty acid, just like in as you can see in sphingospholipids, there is an attached fatty acid. But the difference is that it may have a monosaccharide or an oligosaccharide attached to it. So that is what makes this glycolipids unique or different from this sphingophospholipid. So its role as a component of biomembranes, especially abundant in chloroplast membrane. So that is a curious one, chloroplast membrane. So that is a point to be remembered. And component of myelin sheath, that also important, especially cerebroside. So cerebroside and ganglioside are two examples. So the first one I mentioned, the presence in myelin sheath was ceramide, and apart from that, cerebroside also was present in myelin sheath. So first of all, cerebroside, the structure. So you can compare the structure of cerebroside and ganglioside here. So both of them have sphingosine here, and here it's the fatty acid. This is the same fatty acid here also, but they differ here. Whereas in cerebroside, this carbon will have an attached galactose or glucose. So depending upon that, it may be galactocerebroside or glucocerebroside. But as for here in ganglioside, it will be replaced by a complex oligosaccharide. So that is the difference of cerebroside from ganglioside. Let us see what cerebroside does in our body. Now, cerebroside is abundantly seen in white matter and myelin sheath. Monosaccharide mostly is galactose, sometimes glucose also. So this is galactolipid. It can be said to be galactolipid. Now, different cerebrosides differ in the type of fatty acid part. Obviously, the fatty acid can be different in different cerebrosides, as in the case of phospholipids also, as also in the case of triglycerides also. If the fatty acids are different, the name or the behavior also changes. And then different cerebrosides. Gaucher's disease is an inborn error of lipid metabolism in which cerebrosides accumulate in liver. So that is something clinical. Gaucher's disease, with accumulation of cerebroside. So like in the case of ganglioside also, so there are also Tay-Sachs diseases there. It's an inborn error of lipid metabolism where ganglioside accumulate in brain, whereas here in cerebroside it was accumulated in liver. Gaucher's disease, it is in liver, but in ganglioside it is accumulation in brain. In Tay-Sachs disease, the ganglioside accumulates in brain. Now, ganglioside obviously formed of galactose, glucose, sialic acid, hexamine. So this forms the oligosaccharide, and there are hexasamine, and this forms the basic oligosaccharide, and there is obvious ceramide portion which is having fatty acid and sphinginin. Now, the reason why I say ceramide portion is because this much is the same as ceramide. This much is the same as ceramide. Now, cerebral cortex is rich in gangliosides. So, brain contains gangliosides. Now, we move on to derived lipids. So, now we move over to derived lipids. Now, derived lipids mainly include terpenes and steroids. And these terpenes and steroids are called isoprenoids because they contain what is called isoprene units in them. So this is isoprene unit, 5 carbon number isoprene unit. But these compounds do not arise from isoprene itself, or isoprene is not a precursor of them. Instead, they are formed from precursors to isoprene units in biological systems, namely isopentenyl pyrophosphate and dimethyl allyl pyrophosphate. Now, examples of isoprenoids include carotene, phytol, retinol, vitamin A, tocopherol, vitamin E, squalene, and others. And lanosterol and cholesterol, they are also derived from squalene, and squalene is obviously it's an isoprene derivative. Now terpenes. Terpenes are phytochemicals commonly produced by plants as secondary metabolites. And these secondary metabolites are responsible for some of the special products of plants, like which produces fragrance or essential oils, resins. And the list is actually, it's a long list. Now, terpenoids are terpene derivatives, like there are examples given here: camphor, menthol, carotene, and a number of terpenoids can be added to this list. No. So, are they made by joining isoprene molecules together? No. The process actually involves the isoprenoid pathway, as I said, starting from isopentenyl pyrophosphate and dimethyl allyl pyrophosphate. So, free isoprene molecules are not involved in the process, but we can detect isoprene units in them. For example, the monoterpenes have 2 isoprene units in them, sesquiterpenes have 3, diterpenes have 4, and so on. Generally, isoprene, since it is C5H8, so terpenes are C5H8N times. For example, this is shown here. Menthol from mint plant, it's a monoterpene having 2 isoprene units. Zingiberine is from ginger, it's having 3 isoprene units in them. So this is these are isoprenoids. Now the second category, namely fuck that. Now, the third category, steroids. A typical member, cholesterol of steroids is shown here. The structure is here. We can examine the special features of steroids using this structure of cholesterol. Now, saturated polycyclic hydrocarbons, there are saturated polycyclic hydrocarbons in this structure. Cyclic derivatives of terpene, isoprene subunits, can be detected in cholesterol. Contain fused cycles are here. Contain cyclohexane is there, here. Cyclopentane, one cyclopentane is also there in cholesterol structure. Contains mostly OH groups. OH group is here, hydroxyl or alcohol group is there, and that's why they are called sterols also. Steroids are called sterols because of the presence of OH group. No fatty acids, so there is no fatty acid attached, but of course, cholesterol or other steroids can form esters by attaching to fatty acids. There is no glycerol or special alcohols attached. Now, sterols are present in eukaryotes only, so they can be of plant origin or animal origin, and the derivatives, and this is often described as the derivatives of cyclopentanoperhydrophenanthrene ring system. And as I said, soosterols of animal origin, phytosterols of plant origin, then yeast origin as ergosterol, and cholesterol then. Now, cholesterol is abundant in animal tissue in free state or as esters attached to fatty acids. They are, regarding function, they are cell membrane structural component along with the integral member of cell membrane along with phospholipid, fatty acids and others. And they form a precursor of a handful of molecules as being shown here. They actually on breakdown, they give rise to bile acids, bile salts, bile alcohol, and bile salts, especially the breakdown product, the excess cholesterol is broken down to bile salts finally, which are released to the intestine, so they act as for emulsification for the digestion of lipids, so that is the role. Then sex hormones like testosterone, estrogen, they have cholesterol as precursor. And cholesterol is also the precursor of other steroid hormones like cortisol and cortisone. Then cholesterol is further, again, the precursor of a vitamin, the vitamin. Now, let us try to draw the structure of a cholesterol molecule. I'm going to tell you a story of a few brothers. The first one is here, represented by a hexacycle, and his sibling is here, represented by another hexacycle. And these two were not very popular in their locality. One of them was a drunkard, so we assign him an OH group representing an alcohol group. And the second one is a gangster, a thug, he had an extra bond. So that is represented by this unsaturated double bond, unsaturation there. And their cousin, he is very popular, very nice person. He is very much liked by the people there. And as happens to nice persons, they get married very quickly. And he is married to his lady love, who is represented here with a pentacycle. So this couple were staying in a house numbered 18. The brothers were staying in a house numbered 19, and we can expect the married family to branch out. What happens now? Yeah. Children, and children. What's the number? 20. You have to remember this point, that is the 20th carbon. Now, children 21, 22, 23, 24, 25, and twins 26 and 27. So, where does the first carbon start from? It's from here, So, it goes like this, this is the 17th carbon. And so, this is the structure of cholesterol. Cyclopentenoperhydrophenanthrin ring system.